ACT Science Tutoring Packet, Answer Key Part 2. So, Part 1, we talked about the test. We did a practice section passage, and then we talked a little bit about charts and graphs and what's to be expected. Again, you're going to need pause because I'm going to go straight to the answers here. And it's a little tough because I've only got a computer screen. I can't zoom out so far because you won't be able to read it. But this is a drill. It's not a whole passage, so it should not take you nearly as long. And it talks about the kinetic molecular theory, providing new insights, blah, blah, blah. This is a great example of a passage that I just truly don't understand in terms of really at higher temperature it has a lower number of molecules in motion. That seems odd to me, but maybe they're volumetrically identical. Maybe it means the water sample's a little different. That's what's going on. Doesn't matter. As long as we can answer the questions, all is good. So again, pause it, read the questions, check out the graph, and I'm going to go through them. So number 1, which of the following statements best describes the changes observed in the graph? So this is just cross them out as you go along. T1, sample 1 has a lower kinetic energy than does sample 2. At temperature 1, no, it has a higher kinetic energy, using the number of molecules to proxy that. At T2, sample 1 has a lower kinetic energy than does sample 2. That's correct, so I'm going to assume that's the answer. An increase in temperature leads to decrease in kinetic energy. That would seem to be the case in that number of molecules in motion has gone down, but no, that's not quite the case. Increase in temperature increases kinetic energy. So I said I don't quite understand this. It's got a lower number of molecules at its peak, but it's got more in terms of a fatter area under the curve. So it's real tricky. And water never undergoes phase change. You always find that on the ACT. One answer that's just kind of ludicrous. Actually, this makes no sense at all. Increase in temperature leads to a decrease in kinetic energy. It's got to go up. Number 2. Assume that water undergoes a phase change to a gas, which the following statements would be true. So we look at this and we say the attractive intermolecular forces of the escaping molecules are weak. And I say, yeah, that's it, because of this sentence. The average kinetic energy of the water remains the same. No, it's changing as it gets hotter. Rate of movement of the gas molecules decreases. No, they're going to increase more. Water never, another nonsense answer. 3. Suppose a third sample of water is heated to a higher temperature. It is then found that a greater amount of molecules escape the liquids in sample 3 than sample 2. Would this result be consistent with the results depicted in the figure? Yes, because of this higher energy here than the little energy below it. So it's yes. Get rid of the no's. Don't even deal with them. It's very typical. Yes, yes, no, no. Agree, agree, disagree, disagree, that sort of thing. Yes, an increase in temperature leads to a decrease in no. Yes, as the temperature increases, it leads to more molecules escaping the liquid. Does that make sense? And there is a little bit of prior knowledge here, but if you read it carefully, you should be good to go. It can be inferred from the passage that when a substance undergoes a phase change from a liquid to a gas, it will. This feels like prior knowledge to me. Do you know? Remain the same, doesn't make sense. Disintegrate, I don't know, we're not going to blow it up. Condense means turn from gas to water. Evaporate means change from water to gas. Given the samples T1 and T2, the kinetic energy is measured and shown in the figure. Which temperature has produced the highest single one? T1. How did I know that? T1. Right there. Yeah. Now the passage, the drill I should say. Go ahead and read it. Pause it. Look at your chart. When you're done, take a second and say, what's going on here? Putting a bunch of tadpoles in this hormone, thyroxine, which seems kind of cruel, but whatever, and it makes them mature faster. It makes them go through metamorphosis quicker. The higher the concentration, the faster they go. So, let's answer some questions. Suppose tadpoles immersed in 0.3. We don't have 0.3 for 72 hours. 72 here is right about here. 0.5, 0.2, 0.3 would actually be a little bit lower than that. So we're looking at about 31, 32. Only one close to that is 30%. So 1 is B. Which of the following generalizations works? Temperature plays I'm looking at J. Temperature plays a major role. I like to find the nonsense when I toss it out. They never said anything about temperature. They will not undergo metamorphosis if they're not given thyroxine. Well, tadpoles go undergo metamorphosis all the time, so that makes no sense. Metamorphosis in a normal tadpole takes at least 5 days. I don't know, I suppose so. I really don't know. The most rapid disappearance of the tail is associated with the immersion in the most dilute. No. The most rapid disappearance is the one that's least dilute. Here we have 0, 1 day, 2 day, 1 days, 2 days, 3 days, 4 days, 5 days, and that's the control group that we haven't done anything to, and it hasn't lost its tail completely. So that's what they're saying here. It takes at least 5 days. Gotcha. After 4 days, the tadpoles are checked for development, and all samples other than the control, which the following concentration would the tadpoles be likely to show the least development. So what they're doing with these least and greatest and more, they're looking for double negatives to cross you up. Which one's going to have the least development? It's going to be the lowest one. Now you would think it's this one, but they said other than the control. So you're looking at 0.1. Making sure I put the right answers down. Yes, I did. Okay. 4. Based on the information passage, which the following would be a correct order of the stages of tadpole development. I'm bothered that I don't have that done right. Order of the stages of tadpole development. This one bothers me, because if you read the passage, it says determination of cells. Where is it? It begins with the determination of cells, but if you read the whole thing, at the tadpole stage, so if you don't have a tadpole, you can't have determination of cells. By the way, what does determination of cells mean? I have no idea. So it's not that one. It's tadpole cell determination. It's this one. Reabsorption of tail in adult. That one's tricky. They're doing their best to mess with you. According to the figure, immersing the tadpoles in solution does not begin to affect the rate until when? So you're looking at the chart saying, when do they start branching? There's no branching here. That's 12 hours. So it did not affect them until after 12 hours. Alright. Another drill. I'm not going to answer any of these. I'm going to let you play with it. Pause it, read it, and then I'm just going to put the answers up. This one tricks people. You got to notice that this is going up, this is going down, so they're inverse, so you're looking at A or B. This one has a bigger gap here than here, so it's got a curve to it. Watch out. I guess I will go through them a little bit. So what conclusion could be appropriately drawn from the date regarding electronegativity? Should be data. An element with high electronegativity is a nonmetal. Three, what generalization can one make concerning the relationship between the two properties of elements? This is what I'm talking about. Atomic radius, electronegativity are opposite, so no. As the atomic radius decreases, electronegativity increases. That looks good to me. All metals have higher than nonmetals. Nope. Metals are up here, nonmetals are down here. Atomic radius of F is larger than that of N. Atomic radius, no. So just go through them. Which following is true regarding the comparative electronegativity of fluorine and lithium? Fluorine is variant electronegativity and Li is not. F is greater than Li. F is greater than Li. Li is greater, no. So we're looking at one of these. F electrons are more tightly bound. F is fewer. Now we gotta go back to the reading and check it out. Turns out this. Higher electronegativity comes from more tightly bound. And this happens a lot where you have to go find those. It's a measure of the relative strength with which the atoms attract outer electrons. So they're more tightly bound, they attract more electrons. It says it somewhere in here. Yeah. This is ballpark prior knowledge, so you gotta be careful. But at least you should be able to get it down F and G. Generally speaking, ionization energy follows the same trends as does negativity. So, it should be higher or lower we go on the chart. So we should have F at the end. So, definitely not Li. Definitely not Li. PNBE could be it. Nope. Goes B then N then BE. So let's check to make sure. LINF. Yep, that'll work. Here is a practice problem to try. It's going to be tough. You have to pause it. I'm just going to post answers. I will just post answers on this one. I'm not going to actually walk through the whole thing. So pause it, read it, and there's a whole stack. This is a real passage. We talk about human development. I'm going to give you a big hint because I like you. Birth is right here. Boy, did that mess with me. And by the way, I have no idea. I've never taken biology in my life. I have no idea what they're talking about there. So again, I'm just going to slap some answers up. All right. Some complicated looking questions, but if you keep your head about you, it's not too bad. So that was a real passage. This is also a real passage. All right, they're talking about a parallel plate capacitor, which I actually know something about, but it doesn't mean I can answer the question easily. I had to work really hard to answer this one. So here's all your data. Then you answer the questions. And again, I'm just putting up answers. This one's pretty nasty. Lots of units, lots going on there. There's 1,000 millimeters in a meter. Helps if you know that. If you don't, yeah, you definitely got problems. And that will do it for this one. We will talk about experiments in our next part. Thanks for listening.